Hello guys, this is Rasagna and welcome to my YouTube channel. This is my first video regarding an assignment in genetic engineering and I'm gonna explain about cloning enzymes. The cloning enzymes are very essential in the domains of industrial biotechnology and genetic engineering. The process of cloning enzymes typically begins with the identification and isolation of the gene responsible for producing the desired enzyme. This gene is then inserted into a suitable vector such as plasmid or a viral genome, which acts as a carrier for the gene. The vector containing the gene is then introduced into host cells such as bacteria, yeast, or mammalian cells through a technique called transformation. The host cells serve as factories for enzyme production, utilizing their own cellular machinery to synthesize the enzyme of interest. The cloning enzymes are of 8 types. They are nucleases, polymerases, ligases, transferases, DNases, RNases, kinases, and phosphatases. Nucleases are a group of enzymes that play a critical role in DNA and RNA metabolism. Nucleases are divided into exonucleases and endonucleases based on their function of cutting the DNA. Exonucleases are the nucleases which cut the DNA strand from the ends. They are of two types: lambda exo, which cuts the DNA at 5' end, and exo 3, which cuts the DNA at 3' end. And on the other side, endonucleases cut both single-stranded DNA and double-stranded DNA, but at the middle of the DNA strand. Some of the endonucleases are S1 nuclease, Mung bean nuclease, and etc. The function of polymerases is to form a phosphodiester bond within a DNA molecule. Generally, polymerases have a tendency to make mistake for every 10^6 nucleotides. This is called error-prone or fidelity of replication. Taq polymerase is one of the famous polymerases as it could resist high range of temperature, approximately till 95 degrees Celsius. There are even other types of polymerases such as DNA polymerases, RNA polymerases, viral polymerases like T4 or T7 polymerases. And some of the other polymerases are like RT polymerases, which help in reverse transcription process. Ligases are enzymes that catalyze the formation of covalent bonds between molecules. Ligases tend to ligate the phosphodiester bonds between two DNA molecules. DNA ligases are essential for DNA replication, repair, and recombination. They are of two types based on their source of extraction. They are DNA ligases extracted from E. coli and T4 DNA ligases extracted from T4 phase. RNA ligases are involved in RNA processing and modification. They play a crucial role in the maturation of tRNA molecules. Transferases in cloning add nucleotides only towards the 3' end of the DNA molecule. It is actually termed as terminal deoxytransferase. It is defined as an enzyme which catalyzes the transfer of a particular group from one molecule to another. An example is peptidyl transferase. Transferases play a fundamental role in cellular processes and are of great interest in biotechnology and medicine. Understanding their mechanism and functions can provide insight into disease mechanisms and enable to develop tools for their control. DNases, also known as deoxyribonucleases, are a class of enzymes that catalyze the hydrolysis of DNA molecules by breaking down the phosphodiester bonds that connect nucleotides. They are involved in various biological processes including DNA degradation, DNA repair, and gene regulation. DNases have important applications in molecular biology, diagnostics, and potential therapeutic interventions. They are of three types, namely DNase 1, DNase 2, and MNase. It is also used in the process of RNA isolation if DNA acts as a contaminant. RNases, also known as ribonucleases, are a class of enzymes that catalyze the degradation of RNA molecules by cleaving phosphodiester bonds between nucleotides. They participate in the turnover of both mRNA, which carries genetic information from DNA to the protein synthesis machinery, and non-coding RNA molecules involved in regulatory functions. They are of three types: RNase A, which is specific towards adenine and cytosine nucleotides during degradation, and RNase C, which is specific towards uracil nucleotide during degradation. RNase H helps in denaturation and unwinding of RNA DNA hybrids. Kinases are a class of enzymes that play a central role in cellular signaling and regulation. They are responsible for the transfer of a phosphate group from adenosine triphosphate, that is ATP, to a substrate molecule, a process known as phosphorylation. They are key regulators of cellular signaling pathways, controlling a wide range of cellular processes. The dysregulation of kinases can lead to diseases, and targeting kinases has emerged as an important strategy in drug development. Phosphatases are a class of enzymes that catalyze the removal of phosphate groups from molecules through a process called dephosphorylation, mostly in cloning process. Phosphatases act on a variety of substrates including proteins, lipids, and nucleotides. Protein phosphatases are the most well-known and extensively studied phosphatases. They target phosphorylated amino acid residues such as serine, threonine, and tyrosine on protein substrates, effectively reversing the signaling events initiated by kinases. The study of phosphatases and their functions offer potential avenues for therapeutic interventions. So, these are some of the insights of cloning enzymes used in cloning process. I hope you like this video. Please do like, share, comment and subscribe to my YouTube channel. Thank you.